Imagine an ongoing massive battle. You have these frontline troops, and they're fighting incredibly hard, taking heavy fire in the trenches.
Right.
But if you look behind the lines, their main training base, the facility that's supposed to be, churning out specialized reinforcements, has mysteriously shut down.
Just completely closed up.
Exactly. The doors are locked, the lights are off, the drill sergeants are just gone. Yet somehow, when you look back at the front lines, these elite, highly trained veterans are still showing up to fight.
Which doesn't make any sense logistically.
Right. How is that physically possible? Where are they coming from if the base is closed?
It sounds like, the setup to a war movie, but it's actually a profound biological mystery that is happening right now on a microscopic level.
Yeah.
It perfectly describes the immune system of patients who are battling high-grade serous ovarian cancer.
And that is exactly what we are unpacking today. Welcome to the Deep Dive.
Glad to be here.
So we've got our hands on this really groundbreaking 2026 research paper from the journal Immunity, published by Nathan and colleagues.
It's a fantastic paper.
It really is. And our mission for this deep dive is to map out the bizarre cellular geography of this exact battlefield. For you listening, we are going to meticulously break down how your immune system's B cells operate, why these vital local training bases just inexplicably shut down, and how they surprisingly act as a hidden reservoir.
A completely hidden reservoir for cancer-fighting memory cells.
Exactly. So let's jump right in, set the stage for us.
Well, to set the stage, we first need to understand what a quote-unquote good immune response to cancer actually looks like.
Okay.
When oncologists see B cells actually infiltrating an ovarian tumor, it's generally associated with a much better prognosis.
Because it means the body is actually fighting back.
Right. It means the patient's adaptive immune system recognizes the tumor as a threat and is actively trying to kill it.
Makes sense.
Now, the standard textbook assumption in immunology has always been that these frontline B cells are trained and deployed from the local immune bases, what we call the nearby tumor-draining lymph nodes, or TDLNs.
Okay, let's unpack this concept of a training base for a second. Because in a healthy, normal immune response, a lymph node forms this specific microanatomical structure called a germinal center.
Yes, the GC.
Right. And I always think of a germinal center as a mix between, like, a military boot camp and a high-tech weapons R&D lab.
That's a really good analogy, actually.
It's this dedicated zone inside the lymph node where B cells rapidly multiply when they detect an invader. But they don't just multiply, right? They mutate.
Yeah, that is the most fascinating part of the process. It's a mechanism called somatic hypermutation.
Somatic hypermutation. Explain that.
So as the B cells rapidly divide, they intentionally scramble the specific section of their DNA that codes for their antibodies.
They're literally mutating their own DNA on purpose.
Exactly. They're basically playing an evolutionary lottery in real time.
Okay.
They mutate over and over to see who can create an antibody that binds most perfectly to the target.
And when one of them gets it right...
Once a B cell hits the jackpot and creates a highly precise antibody, it's selected to survive, it multiplies further, and it becomes what we call an antibody-secreting plasma cell.
So it graduates boot camp and becomes the heavy artillery.
Precisely. It's an incredible biological machine.
Right.
So naturally, the researchers wanted to see this boot camp in action.
Right. They wanted to catch it in the act.
Yeah. They took fresh, surgically removed tumor-draining lymph nodes and matched them with the primary ovarian tumors from the exact same patients.
Oh, wow. So they had paired samples.
Yes, paired samples, which is crucial. They wanted to look inside these nodes and literally watch the training happen.
And to do that, they didn't just use a standard microscope, right? They used some heavy-hitting tech. Let's talk about seq-RNA-seq.
Right, single-cell RNA sequencing.
Walk us through how that works because it's super important for this paper.
Basically, cRNA-seq isolates individual cells and reads their active genetic diary.
Their genetic diary. I like that.
Yeah, it lets you see exactly which messenger RNA transcripts are present, which tells you exactly which genes are turned on at that exact moment.
So they were looking for the specific genetic signatures that scream, hey, I'm an active germinal center.
Exactly. And the other tool they used was flow cytometry.
Flow cytometry.
Right. That lets them scan the specific protein uniforms on the surface of thousands of cells per second using lasers.
Lasers. Awesome. So they go in with these advanced tools fully expecting to find a buzzing, active germinal center full of B cells mutating and, preparing for war against the ovarian tumor.
That was the expectation.
But what did they actually find?
A complete ghost town.
A ghost town.
Yeah. The entire active training machinery was just gone. When they looked at these tumor-draining lymph nodes, there were virtually no active germinal center B cells. There were no antibody-secreting plasma cells, those fully trained artillery units we talked about. They even looked for the T follicular helper cells.
Wait, what are T follicular helper cells?
Think of them as the drill sergeants. They're the specific T cells required to run and organize the germinal center. And those were completely missing too.
Wait. If lymph nodes are literally built from the ground up to host these germinal centers, why is the local machinery completely switched off right next to a massive, dangerous tumor?
It's a great question.
Is the patient's whole immune system just broken from the strain of the cancer? Like, is their body just giving up?
That would be the logical assumption, right? But data showed something much more specific. It's not a systemic body-wide failure at all.
It's not.
No, it's a highly localized suppression. And to prove this, the researchers brought in control groups.
Okay.
They compared these ovarian cancer lymph nodes to tonsils from pediatric patients, as well as lymph nodes from patients with inflammatory bowel disease, or IBD.
And I'm assuming those control groups looked completely different.
Night and day. The tonsils and the inflammatory bowel disease nodes were absolutely packed with active, thriving germinal centers.
Wow, okay.
That proved unequivocally that the ovarian tumor-draining lymph nodes were uniquely compromised. The local environment, right there next to the tumor, simply could not support the active training process.
Okay, this is a perfect time for a quick recap for you listening, because we've covered a lot of ground in just a few minutes. Let's bullet point this.
Sounds good.
Number 1, B cells fighting inside an ovarian tumor is a good thing for the patient. Number 2, these B cells are supposed to be trained in the nearby tumor-draining lymph nodes inside structures called germinal centers.
Right. The boot camps.
The boot camps.
Okay.
And number 3, using advanced genetic and laser scanning, researchers found that in ovarian cancer, these specific boot camps are basically shut down. No recruits, no drill sergeants, nothing.
Exactly. The training base is closed.
But that leaves a massive hole in the logic.
It really does.
If the base is closed, where are these veteran B cells that are inside the tumor actually coming from?
That mystery drove the researchers to the next phase of the study. They basically had to shift their focus.
Like looking for a different clue.
Yeah. Instead of looking for the active germinal centers that weren't there, they asked, what is actually taking up space inside these lymph nodes?
Because they weren't just physically empty balloons.
Exactly. They were dominated by a completely different kind of cell. They found that roughly 70% of the B cells resting in these nodes were memory B cells.
Memory B cells. Okay, so these are the cells that stick around after an infection to make sure you don't get sick from the same thing twice.
Right.
The paper specifically called them classical class-switched memory B cells. What does class-switched actually mean in this specific context?
Class-switched is a critical detail because it tells us about the cell's history.
Its history?
Yeah. When a B cell is first born in the bone marrow, its default antibody receptor is a basic type, usually an IgM or IgD isotype.
So like a standard issue equipment.
Exactly. This is a general purpose tool. But during that intense germinal center training process we talked about earlier, the mutation lottery, a B cell can literally cut out a piece of its own DNA to switch to a more specialized, powerful antibody class.
Oh, wow. So they cut their own DNA to upgrade?
Yes. They switch to classes like IgG1 or IgA, which have different functional abilities.
So, to use our military analogy, it's like a new recruit trading in their standard issue pistol for a specialized sniper rifle or like a laser-guided missile system.
That is a great way to visualize it. And the fact that these resting memory cells in the lymph node are class-switched proves unequivocally that they have seen action before.
Because they couldn't have that sniper rifle unless they passed the training.
Exactly, and it gets even more definitive. The researchers sequenced these cells and found they carried around 20 somatic hypermutations in their genetic code.
Ah, so that's the smoking gun right there.
Yep.
You only get those specific targeted mutations if you've survived the grueling germinal center boot camp.
It's exactly right. It proves that while these memory cells are currently resting, they are in what we call a quiescent state. They did go through an active training process at some point in the past.
Okay, but couldn't they just be tired, like burned out?
Sure, right? The researchers checked for that too. They looked for known markers of immune exhaustion, like HAVCR1.
HAVCR1. Got it.
Sometimes, in cancer, immune cells just burn out from chronic stimulation. But these memory cells didn't show those exhaustion markers.
Wow.
There were healthy, highly trained veterans just waiting in reserve.
I'm stuck on something, though.
What's that?
You have all these resting veterans hanging out in lymph node, drinking coffee, waiting for the call. But how do we know they even care about the ovarian tumor? Right? Like maybe they are just leftover memory cells from a terrible flu the patient had 5 years ago, and they're just using the lymph node as a hotel.
That is the exact skepticism you need in science. To definitively answer that, the researchers had to test the actual weapons these cells were carrying.
Okay, how do you do that?
They used a technique called VDJ sequencing. It maps the highly variable region of the B cell's DNA, the specific genetic sequence that codes for the very tips of the antibody.
The tips? Why the tips?
Because that tip is the exact part that physically grabs onto an enemy.
Oh, I see.
So the researchers extracted this genetic code from the resting memory cells. They artificially synthesized those exact antibodies in a lab, and then physically washed those cloned antibodies over living, cultured ovarian tumor cells.
That is incredibly cool. And did they react?
The reaction was undeniable. 36% of these cloned antibodies strongly and specifically bound directly to the ovarian tumor cells.
36%, that's a huge chunk.
It is. They were completely tumor-reactive. And through further testing, they even identified one of the specific molecular targets these antibodies were grabbing onto.
Ooh, what was it?
An enzyme called MMP14.
MMP14, the machete. I love the specific detail from the paper.
That's the great visual.
So for you listening, think of MMP14 like a biochemical machete that the tumor uses to hack its way through the body. A growing tumor needs to chop up the extracellular matrix, which is, the structural scaffolding that holds our healthy tissues together so it can spread and metastasize.
Exactly.
MMP14 is that machete sitting right on the surface of the tumor cell. And these resting memory B cells had created specialized antibodies specifically designed to target, bind to, and neutralize that tumor machete.
It's a brilliant, highly specific biological adaptation. But, it does raise a complex issue about how the immune system distinguishes friend from foe.
Right, because I was going to ask, MMP14 is a protein our own bodies produce naturally, isn't it?
It is, yeah.
Our healthy cells need to remodel tissue sometimes, too, so these antibodies are technically self-reactive.
Yeah.
Why isn't this massive reserve army of memory cells causing a horrifying autoimmune crisis all over the patient's body, just like attacking healthy tissue everywhere?
It comes down to the unique pathology of the tumor microenvironment. It's really a matter of concentration and context. Well, while healthy tissue might express tiny, tiny amounts of MMP14 during normal processes, the ovarian tumor has a massive pathological abundance of it. It's highly concentrated on its surface as it aggressively tries to spread. The antibodies are dialed in to react to that overwhelming density.
So they need a strong signal to wake up.
Exactly. They aren't blindly attacking the patient's whole body. Their activation threshold is triggered by the dense concentration found only at the tumor site.
Incredible. Okay, let's pause again for our second quick recap.
Let's do it.
Number 1, the lymph node's training base is shut down, but the node is actually packed with memory B cells. Number 2, these cells are class switched and highly mutated, meaning they're experienced veterans.
Yep.
And number 3, when tested, over a third of these cells produced weapons specifically designed to neutralize the tumor's primary spreading tool, the MMP14 machete.
Spot on.
But that brings us to a massive logistical problem. Having a reserve army in the barracks is great, but how do we prove they are actually waking up, leaving the lymph node, and traveling to the front lines inside the tumor?
You need a physical connection.
Yeah.
You have to prove that the specific cells resting in the node are the exact same lineage of cells doing the fighting in the tumor.
And how do they do that? Because they had paired samples.
Right, exactly. Because they took the lymph node and the tumor from the exact same patient at the exact same time, they could look for what we call a clonal connection.
Here's where the science gets really elegant. We need to talk about the barcode concept, or clonal lineage.
It's a beautiful concept.
Every single B cell, when it's born, creates a completely unique DNA sequence for that variable antibody tip we just talked about. It's essentially a highly specific, unique barcode. If that single B cell divides and multiplies into an army, all of its clones will share that exact same unique barcode. So, if you find the identical barcode in 2 completely different locations in the body, you know with absolute mathematical certainty that those cells share a common ancestor.
That barcode is the key to tracking their migration. Now remember, inside the primary tumor itself, the researchers did find an active immune response.
Oh, right, the fully trained ones.
Yes. They found those terminally differentiated antibody-secreting plasma cells actively fighting right there in the tumor microenvironment.
Okay.
So they ran a massive clonal trajectory analysis. They mapped the barcodes of the active plasma cells fighting in the tumor, and they compared them to the barcodes of the resting memory B cells sitting back in the lymph node.
The suspense is killing me. Did they find a match?
They found perfect matches.
Oh, wow.
The resting memory cells in the lymph node shared identical genetic barcodes with the active plasma cells deep inside the tumor mass.
So let me make sure I'm visualizing this correctly for our listeners.
So,
The lymph node isn't running the factory anymore. The lymph node is essentially just a storage locker, like a barracks.
A holding zone.
And these veteran memory cells are actively migrating out of the barracks, traveling through the body to the tumor, and then finishing their final transformation into active plasma cells right there on the actual battlefield.
If we connect this to the bigger picture, that is exactly what the data suggests.
That's wild.
The tumor microenvironment itself is partially compensating for the broken lymph node. Because the draining lymph node can't train new recruits, the body relies on these memory cells to traffic to the tumor mass and set up shop on the front lines.
So the lymph node acts as a vital physical reservoir supplying the ongoing fight.
Exactly. It's keeping the fight going.
Okay, time for recap number 3. We've solved the mystery of the supply chain. We know where the reinforcements are coming from. The node and the tumor share the exact same genetic barcodes.
Perfect summary.
But we've actively ignored the most glaring, suspenseful question of this entire deep dive.
I know where you're going with this.
If these lymph nodes are naturally supposed to be training centers, who or what sabotaged the germinal centers in the first place? Who closed the base?
Moving from the what to the why required the researchers to look at the physical, spatial architecture of the lymph node tissue.
Like looking at a crime scene map.
Exactly. They needed to see exactly who was hanging around the crime scene. And to do this, they used an incredibly advanced imaging technique called MIBI-TOF.
MIBI-TOF, which stands for multiplexed ion beam imaging by time of flight.
That's a mouthful, I know.
Yeah. Now, I've heard of tagging cells with glowing fluorescent colors to see them under a microscope, but MIBI-TOF is a completely different beast, right? Break that down for us.
It is revolutionary spatial mapping. With traditional fluorescence, you can only use a few colors before they blur together.
Like mixing too much paint.
Exactly. But with MIBI-TOF, instead of glowing colors, they tag the antibodies with rare, heavy metal isotopes.
Heavy metals.
Yeah. Then they shoot an ion beam at the tissue, vaporizing the top layer atom by atom.
You're kidding, they vaporize it.
They do. And a mass spectrometer catches the particles flying off and measures the exact weight of those metals. It allows researchers to map dozens of different cells and proteins in stunning, high-definition 3D at the exact same time.
That sounds like something straight out of science fiction. You're literally painting cells with heavy metals and vaporizing them to see where they live.
It's unbelievable tech.
So when they looked at this high-definition map of the sabotaged lymph node, what did they find at the crime scene?
They found a heavily invading population of macrophages.
Macrophages.
Specifically, a type of macrophage expressing a surface protein called DCCGN. So DCCGN positive macrophages.
Okay, macrophages aren't those usually the immune system's trash collectors? Like, their job is usually to patrol around and eat dead cells or engulf bacteria?
Well, normally, yes.
But here they sound like bouncers who have completely taken over the club and are shutting the whole thing down.
That's exactly what it looked like. The MIBI-TOF imaging showed these macrophages sitting directly inside the microanatomical zones where the B-cell germinal centers were supposed to be.
Just loitering in the boot camp.
Right. And the researchers found a direct inverse correlation. The more of these DCCGN positive macrophages that were present in a patient's lymph node, the fewer germinal centers survived.
But hold on. Seeing them at the scene of the crime doesn't necessarily prove they committed the murder.
Correlation is not causation.
Exactly. How do we know it wasn't just the toxic inflammatory environment of the cancer itself that killed the training base, and the macrophages were just there to clean up the dead body?
That is exactly the question you have to ask in science. So they needed a smoke and gun experiment.
What did they do?
They set up a fascinating co-culture experiment in the lab. They took healthy, active germinal center B cells from tonsil cells that were actively mutating and training and put them in a dish with these specific tumor node macrophages.
And what happened? Did the macrophages secrete some kind of poison? Like, how did they kill them?
To find out exactly how they were doing it, the researchers used 3D live cell confocal microscopy to watch the interaction in real time.
Oh, man. They recorded the murder.
They did. Over the course of 4 hours, they watched the macrophages physically approach the active germinal center B cells, engulf them, and eat them alive.
What?
Yeah. It is a process called phagocytosis. They literally swallowed the active training cells intact.
That is terrifying.
And here is the craziest part. They completely ignored the resting memory B cells in the same dish. They only ate the active recruits.
4 hours. So they didn't just assassinate them. They slowly digested the new recruits while ignoring the sleeping veterans.
Exactly.
That is both a biological marvel and incredibly grim. And I assume they ran a control to prove it was physical contact and not like a poison they were spitting out.
They did. They used a Transwell assay.
Explain that for the listener.
Basically, they put a physical microscopic filter between the B cells and the macrophages in the dish.
Okay.
The pores in the filter are small enough to let fluids and chemicals pass through, but too small for whole cells to cross.
So it's a physical barrier.
Exactly. Yeah. When they put that physical barrier in place, the B cells survived perfectly fine. It definitively proved the macrophages had to physically touch, grab, and eat the B cells to destroy them.
So the saboteurs are literally eating the recruits before they can finish training.
Yeah.
Why? Why would our own immune system turn on itself like that? That doesn't make any evolutionary sense.
This raises a really important question regarding how our bodies handle chronic inflammation versus acute infection.
Okay, break that down.
These macrophages are normally recruited to lymph nodes to clear out what they perceive as an acute danger, like a severe, overwhelming bacterial infection, where the body needs to quickly clean up cellular debris to prevent sepsis.
Right, an emergency cleanup crew.
But in the context of chronic cancer inflammation, their programming goes haywire.
Yeah.
It becomes deeply maladaptive.
They get confused.
Exactly. They perceive the rapidly dividing B cells in the germinal center as part of the chaotic problem, so they eat them. In their misguided attempt to restore order, they actively suppress the body's ability to generate new, long-lasting adaptive immunity.
It's a tragic misunderstanding at a cellular level.
It really is.
Wow. So let's bring this all together. What does this all mean? Why should you, listening right now, care about the specific geography of a single lymph node?
It's a huge paradigm shift.
Right, because think about how we currently treat this disease. In standard surgeries for ovarian cancer, doctors will routinely remove the tumor-draining lymph nodes.
Standard of care.
The logic has always been to try and prevent the cancer from using those nodes as a highway to spread to the rest of the body. But this paper completely challenges how we view those nodes.
Fundamental.
If these nodes aren't just useless deadweight, if they're actually the vital protective barracks holding the sleeping veteran memory B cells that continuously feed the ongoing fight inside the tumor, then surgically removing all of them might inadvertently weaken the patient's own immune response.
We might literally be throwing away the reinforcements.
Throwing away the reinforcements.
Exactly. It fundamentally reframes our entire therapeutic approach. We spend billions of dollars and countless hours in labs trying to engineer complex, new, artificial immune cells to put into patients.
Like CAR-RT and things like that.
Right. But what if the ultimate cancer therapy isn't building new cells from scratch?
What if it's not?
What if it's simply stopping these macrophages from eating the ones we already have?
Oh, wow.
Imagine if we could develop a targeted drug to just turn off or repel those specific saboteur macrophages. We could protect the training base, reactivate those dormant memory reservoirs in the lymph nodes, and just let the body's own highly trained veterans finish the fight.
It's an incredibly empowering thought for the future of medicine. So the next time you think about the frontline battle against cancer, remember that sometimes the key to victory isn't just focusing all our firepower on the trenches. It's about unlocking the barracks, waking up the veterans, and getting the saboteurs out of the way.
Well said.
Thanks for joining us on this deep dive. Keep questioning the consensus and keep learning.